My name is Kyle Kwan, and this is my protein project for the Biology 101 class of the fall 2020 semester. The protein I had for this project was hemoglobin, and the mutation I was researching was the sickle cell mutation. With this, we get into the normal function of the hemoglobin protein, the wild type hemoglobin protein, which is the fact that one, it gives red blood cells their color, and two, which is its more important function, it allows red blood cells to pick up oxygen in the lungs and allows it to transport it across the body, like to all the different tissues, muscles, and organs through the circulatory system. With this, I deduced that the transport molecule, or the hemoglobin protein, is a transport molecule. This protein, once again, is most commonly found in the red blood cells of animals, and the type of mutation that occurs for it to become for the red blood cell to become a sickle red blood cell is in the hemoglobin. There's a point mutation, or more specifically an amino acid substitution, in which the normal glutamic acid is replaced or taken out and replaced by the valine amino acid. This substitution occurs on position 6 of the beta globin subunit of hemoglobin. And with this, I can show you my my model, which is as you see here, that we have like the alpha and the beta chain of the hemoglobin molecule, and with these little green things inside are the heme groups. The glutamic acid, which its main functional groups consist of the carbonyl and carboxyl group, is usually charged hydrophilic and polar, which is also usually charged hydrophilic and polar, is changed to the hydrophobic and nonpolar valine amino acid. This doesn't really change like the function of hemoglobin, or I guess it does in the sense where it alters it, to which hemoglobin, which usually doesn't stick together within the red blood cells, ends up causing the hemoglobin becomes less soluble under decreasing oxygen conditions, and it starts to polymerize into crystals, which is shown by this. This is my mutated hemoglobin model. So as you see, this is a chain of hemoglobin molecules or hemoglobin proteins. And what this does is it distorts the normal shape of the red blood cell, causing it, and most commonly, to become sickle shaped. Doesn't have to be sickle shaped, but it can also just be a bunch of irregular shapes. But what we know it for is causing the sickle cells, like sickle red blood cell shape. So what this causes within the red blood cell is it causes the red blood cell to lose a lot of its elasticity. The red blood cell essentially becomes rigid. And this is an issue because when the red blood cell is going through capillaries, it needs that elasticity to be able to fit through those small capillaries, or else it would break them. So with the new rigid irregular shape, the capillaries break when the sickle red blood cells are traveling through them, which causes tissue hypoxia. And what tissue hypoxia causes within the carrier is often a lot of pain and a lot of fatigue due to the lack of oxygen being delivered to the places that it's needed. However, the sickle cell mutation isn't all bad, as it gives the carriers resistance to malaria due to the parasites causing malaria being killed inside sickle-shaped blood cells. The limitations to my model include, one, the fact that I wasn't able to show like the specific sequence in which the specific sequence or location where the mutation occurs. Two, I wasn't able to show the effect of this mutation like physically on a red blood cell. And three, being me not being able to directly show a mutated individual hemoglobin molecule. However, with this is the end of my video. Thank you for watching.